Okay, so now at long last we are on to our last type of polymer, the nucleic acids. Phew. As with the proteins, this is a group that we're going to get to know very well over the course of the semester. So at the moment, we're going to focus mainly on the structure of the nucleic acids and how the nucleic acids are organized. We're going to come back to talking about where the nucleic acids are found and their functions inside of cells later on in the course. So mercifully, this video will be on the shorter side. So, what makes a nucleic acid a nucleic acid? So these molecules have a couple of moving parts. Okay, so this thing has a couple different moving parts. So to start out with, let's just talk about what's right here, which is something that you might be able to identify if you squint at this for long enough. And now I'll tell you, so this here is a ribose sugar. Okay, and so notice it has this hydroxyl group in this position here. And we're going to number these carbons, and we're going to start at the carbon that's closest to this thing I've labeled as a base. So that carbon's carbon number 1, and it's indicated with the prime symbol. So 2 prime, 3 prime, 4 prime, and 5 prime. Okay. And right, so if this hydroxyl group were just a hydrogen, then this would be a deoxyribose sugar, right? Okay, so now that I've labeled all these carbons, numbered them, so up here what we've got is a phosphate group, right? So that's a familiar friend. And then now we should come back to talk about this base up here. And this is also often referred to as a nitrogenous base. And you'll see why in a moment. And that base group can come in one of 5 different flavors. So let's take a look at those next. Okay, so now these top 2 flavors up here, you'll notice each have 2 rings. And so these 2 molecules up here are referred to as the purines. And these purines are specifically known as adenine and guanine. And the way that I remember the purines is that it is pure agony to try and remember them. And then these other 3 nitrogenous bases down here each just have 1 ring, and these are called the pyrimidines. Okay, and these are named cytosine, uracil, and thymine. Okay. So, given a nucleic acid or a nucleotide, a given nucleic acid can have any one of these nitrogenous bases attached to it. Well, sort of. So because reasons for DNA, the deoxyribose sugar, DNA can have thymine or cytosine attached, as well as adenine and guanine. Right. Okay, but DNA cannot have uracil attached. And meanwhile, RNA can have cytosine or uracil attached, but not thymine, and it can also have adenine and guanine attached. Okay. All right, so that's kind of a tour of our moving pieces. So the next question is, how are these nucleotides organized? And as with the amino acids, we're going to see there's kind of two major components to this organization. And the first major component involves the formation of a backbone. And in this case, it's a carbon phosphate backbone. So let's draw a picture of that out next. Okay, so one other thing to notice here is that also, as with an amino acid backbone, this backbone is also directional. And so there's an end where the carbon that is free is a 3' carbon. So that's referred to as being the 3' end. And then there's an end where there is a free 5' carbon. So that is referred to as the 5' end. Okay? And this labeling scheme, which orients you to the direction of this DNA molecule, is going to wind up being important down the road. So I think it's helpful to just kind of see where that comes from now and understand why that is the way that it is. So that's a linear component to the organization. And we can keep building one of these chains of nucleic acids and make it longer and longer. And that's actually basically where the story stops for RNA. So if we wanted to write out a molecule of RNA, one of the shorthand methods that's used to do that is we'll start over here with the 5' end. And so in this case, it's going to be RNA. And for a shorthand, we're just going to write out this sequence of bases that's connected together along that chain. So it might be something like A, U, C, G, C, C, A, C, U, G, G, A, dot, dot, dot. And then here's our 3' end, okay? So for RNA, a molecule of RNA is just going to be a single long linear sequence of adenines, guanines, cytosines, and uracils. And notice here that the order of the bases in this RNA molecule can vary, and it doesn't matter what's at position 1. You can have any of the 4 bases at position 2 here. And so that's for RNA. And for DNA, there's one additional component to the organization, and that is that the nitrogenous bases can pair up with each other, and they pair up in a very specific manner. And specifically, purines pair with pyrimidines. And so for DNA, A pairs with T, and C pairs with G. Okay? And then this is going to show up down the road. RNA can pair up with DNA, and in RNA, A pairs with U, and C pairs with G. But we're going to come back to that later on. Okay, so in each case, a purine is pairing up with a pyrimidine. If that was not enough, there is one other twist as well. And the additional twist is that when in DNA the nitrogenous bases pair up, they pair up with the molecule's carbon phosphate backbones running in opposite directions. So if I'm going to rewrite this strand of RNA to be DNA, we'll take a look at this pairing. So if this is now DNA, that's going to be thymine. Okay, so the strand that will pair up with this upper strand will have its 3' end here, and it's going to have what's called a complementary sequence. Okay. And just to show this to you, here's another illustration that I found that shows you the full molecular structure of a DNA molecule. So here on the left-hand side, we've got a free 3' carbon at the end of this chain. And so as we go up, we've got a free 5' carbon up here at the other end. And then on the opposite DNA molecule over here, we've got a free 3' end up here, and then a free 5' end down here. And what we've got here is, here's an adenine paired with a thymine. This is taken from another language site. And then we've got a cytosine paired up with a guanine here. And so a thing I want to highlight from this picture is that what you'll notice are the bonds that are holding together one of these carbon phosphate backbones are all covalent bonds, right? So this is all a set of covalent bonds within one of these strands, and same thing within the other strand. However, the bond that holds these nitrogenous bases together are hydrogen bonds. And so this also shows you, so there are 2 hydrogen bonds present between adenine and thymine, and then there are 3 hydrogen bonds that are present between cytosine and guanine. And the reason I want to point that out is because although these hydrogen bonds will help hold together 2 strands of a DNA molecule, they're actually a lot easier to break than all of the covalent bonds that make up the remainder of the DNA molecule. So there are circumstances where the 2 strands can be pulled apart from each other without losing the overall linear structure of the DNA molecule. All right, with that, I hope you've enjoyed our tour of the major types of polymers that are found within cells. So if we actually go back to this starting list, let's look at all the ground that we've covered already. So, we know a fair amount about water and some of the special properties of water. We've gotten to see how glucose molecules and other sugars can connect together to form polysaccharides as an energy source for cells. We've gotten to know about the triglycerides and the saturated and unsaturated fatty acids, another form of energy storage in cells. We've gotten to see the dual personality of the phospholipids and how that dual personality allows those special molecules to form the phospholipid bilayer, or the membrane within a cell. We've started to see the potential molecular diversity that's possible by combining the 20 amino acids into proteins that are organized according to the primary, secondary, tertiary, and often quaternary structure. And now we've been introduced to the 5 nitrogenous bases that make up the molecules of DNA and RNA. So, that's a lot. And now that we've made it this far, we are next going to turn our attention to a higher level of biological organization, the cell. There will be more on that in the next video.